Hello students, the title of today's laboratory module is Study of Plasmolysis and Deplasmolysis. The aim of today's experiment is to study plasmolysis and deplasmolysis in onion cells. Let us first start with a brief introduction of the principle. All living cells maintain their turgidity due to the presence of water inside the cell. Cell membranes help in maintaining homeostasis in cells by controlling the movement of molecules in or out of the cells across the plasma membrane. Movement of molecules across the plasma membrane is required for maintaining cellular functions as well as for the survival of cells. Although some molecules can move freely across the plasma membrane, other molecules require special transport proteins on the membrane for moving across the plasma membrane. The free movement of molecules across the cell membrane without spending any energy by the cell is known as passive transport. The simplest type of passive transport is diffusion, which involves the movement of molecules across a cell membrane from a region of higher concentration to a region of lower concentration. Another common process of passive transport is osmosis, which is the process by which water molecules diffuse across a cell membrane from a region of higher concentration to a region of lower concentration. The phenomenon of osmosis can lead to plasmolysis or deplasmolysis of cells. Plasmolysis is the shrinkage of protoplasm accompanied by visible separation of plasma membrane from the cell walls when cells are immersed in hypertonic solution. Plasmolysis occurs due to exosmosis, which is the movement of water molecule across the cell membrane from the inside of cells or cell sap to the surrounding more concentrated hypertonic medium. On the other hand, deplasmolysis is the reversal of plasmolysis or the restoration of turgidity of the cells when they are kept in a hypotonic solution. Deplasmolysis involves endosmosis or the movement of water molecules from the less concentrated hypotonic solution of the medium to the inside of the cell. Plasmolysis and deplasmolysis of cells can be demonstrated in the lab using simple materials. Plasmolysis can be observed by keeping the cells in a hypertonic solution and then observing them under the microscope. Deplasmolysis can be demonstrated in the same cells by incubating the plasmolyzed cells in a hypotonic solution or simply distill water for some time and observing them under the microscope. For demonstrating the process of plasmolysis and deplasmolysis, plant tissues with pigments are more suitable as the colored cell sap makes the cytoplasm more prominent when observed under the microscope. Therefore, materials such as peels of lower surface of colored leaves or red onion cells are commonly used for such experiments. Since onions are easily available, the present experiment will use peels of red onions to demonstrate plasmolysis and deplasmolysis. Let us now see the materials required for the experimentation. The materials required include onion bulb, Watt's glass, petri dish, slides, cover slips, forceps, brush, microscope and 20% sucrose solution. Let us now see the procedure of the experiment. First, take a red colored fresh onion and remove the old layers. Now take a piece of a soft fresh layer and peel a layer of the red cells by using a forceps. If the layer cannot be peeled easily with the forceps, then fold the onion piece with the red cell surface facing towards you and fold it by pressing with the fingers. The white tissue will be broken, but the red tissue layer will remain intact. Remove the peel of the white tissue with the forceps and a thin film of the red cells will remain. Now cut the thin red film into small pieces. Take a strip of the onion cells with the help of a brush and place it on a microscopic slide with 1 or 2 drops of distilled water and cover the drop of water containing the onion strips with a cover slip. Keep the slide under the 10x objective of a microscope and observe the cells. Now shift the objective of the microscope to 40x and observe the cells. Examine if the cells are intact or not. Take the strip of the red onion cells with intact cells and transfer it on a new slide on a microscopic slide with the help of a brush and add few drops of 20% sucrose solution on it. After 10 minutes, cover it with the cover slip and mount it under the 10x objective of the microscope, and when a clear fill is visible, observe it under the 40x objective. Examine the cytoplasm of the cells and compare it with the cells mounted with distilled water. Now draw out the sucrose solution from the slide by placing a piece of filter paper on one side of the cover slip. Add few drops of distilled water from the opposite side of the cover slip and keep for about 10 minutes. Observe the slide under the microscope again. Record the observation and compare the cells in the first distilled water with the cells in concentrated solution and then the cells transferred to distilled water again. Let us now see the results. When the peel of onion was first kept in distilled water and observed under the microscope, the intact onion cells were turgid. However, when the onion strip was kept in concentrated sucrose solution, which was hypertonic, the cytoplasm underwent shrinkage and was hardly visible. It demonstrates plasmolysis, or the phenomenon of shrinkage of protoplasm when the cells are kept in a concentrated solution or hypertonic medium due to exosmosis or movement of water molecules from its region of higher concentration in the cytoplasm to its region of lower concentration in the external hypertonic medium. Further, when the concentrated sucrose solution was replaced with distilled water again, the cytoplasm of the cells swelled and became turgid again by absorbing water from the surrounding hypotonic medium, leading to deplasmolysis or reversal of plasmolysis. Let us now see the conclusion. The cell membranes of all living cells are selectively permeable and allow only specific molecules to pass through them. Water molecules can diffuse across a cell membrane from a region of its higher concentration to a region of its lower concentration by a process called osmosis. The phenomenon of osmosis can be observed during plasmolysis and deplasmolysis of cells. Plasmolysis can be demonstrated by incubating intact turgid cells in a hypertonic solution, and deplasmolysis of the plasmolyzed cells can be achieved by placing the plasmolyzed cells in a hypertonic solution. Thank you.